All right. Oh, hello, everyone. Someone type a message in the chat if you can hear me. Make sure everything is working okay here. I think so. Yeah. Okay. Looks good. Thank you. Great. So I hope everyone had a good 2 weeks off, and hope you had some, a good opportunity to practice and work on using R for sort of like, let's say, normal lab data, simple stuff. Because today we're going to, increase the level of complexity and talk about single-cell RNA-seq analysis. And I think this is what most people are interested in doing. So, maybe I'm preaching to the choir a little bit, but let me just start off by with a little introduction in terms of why single-cell RNA-seq is important and useful, and why we're using this platform to, analyze it. And, basically, single-cell RNA-seq is more or less a standard assay these days. 5 years ago, it was more or less brand new. At a commercial level, it had been developed beginning around 2015 as a technique to do the droplet-based single-cell RNA-seq, at least. But it's a standard assay now, I would argue. It has a large number of advantages relative to bulk RNA-seq, which apply to most of us working with in vivo models. These, very basically or very simply include the ability to bioinformatically purify your cells. And, whether you're sorting or selecting prior to doing bulk RNA-seq, these are never, 100% effective. And they're not close to 100% effective techniques. What we've learned and I've experienced over the years is that, a small number of contaminating cell types that produce a lot of RNA, and maybe a lot of a specific type of RNA mixed in with the cells that you're interested in, are going to cause problems for your analysis. There's ways to get around that, but I think this is the most straightforward way to use single-cell RNA-seq to isolate your cells and analyze only the ones that you want. It also enables you to eliminate or segregate or quote unquote gate out poor quality cells. So you can identify these by various metrics, which we'll go through, and eliminate them. So that's an advantage as well. You may not have anticipated certain subsets of cells within your data. And let's say you're studying T cells, for example. Well, there may be, in your experiment, a new subpopulation of T cells develops that you're unable to predict, or you wouldn't have thought of, ahead of time, and certainly couldn't think or wouldn't be able to sort for to analyze. So this allows you in a single experiment, the first experiment that you're doing, to identify de novo, without predicting, new cellular subsets within a population of interest that you can characterize and quantify. One thing that is, absolutely not possible in bulk RNA-seq is the unsupervised generation of gene modules. Now, broadly speaking, you could say a gene module is any group of genes or a set of genes that you care to define. The way we're using it here and what we'll go through in our analysis is genes that are co-regulated from cell to cell at a single cell level in the absence of any other defining characteristics of those cells. So it simply starts at cell number 1 and goes to the last cell in order and says what cell or what genes or what groups of genes go up and down together from the first cell to the last cell. And that's not something you can get out of bulk RNA assays. Pseudotime is another thing that you may be interested in, looking at having a snapshot at a dynamic population where cells are in different states along a trajectory. So that's an important thing. One of the limitations, you might say, of single-cell RNA-seq is sequencing depth per cell. This can be overcome using what we call pseudo-bulk analysis. We'll go through this next week. But this is essentially the ability to combine arbitrarily any group of cells and to get their aggregate gene expression profile. So it's as if you were to have complete power to sort those cells and do a bulk RNA assay, RNA-seq assay on only the cells that you're interested in. And so this allows you to combine any group of cells that you want. Could be T cells, could be subsets of T cells or monocytes or whatever you're interested in, and then get a group expression average for those cells. And the last thing I'll point out is the ability to do multimodal experiments with site-seq antibodies to combine RNA-seq and ATAC-seq and various other things. So I think these are basically all the reasons that you should be interested in doing this if you're doing in vivo studies or translational studies on patient samples. So challenges, and the first challenge I'll mention, actually I forgot to put it on here, is cost. These assays do cost a lot of money. We're not going to get into how much and how to get around that and how to deal with that, but that is a challenge. Another challenge is the biological complexity. Once you get one of these data sets, it's not that you get one gene list. You can have sort of an arbitrary number of gene lists to look through and to figure out what it means. And so the time to analyze, even if you're very experienced at it, becomes quite large. Technological barriers, and this is what I want to, this is the motivation for a lot of what we're doing with this course, is to reduce the technological barriers so that, you're not trying to figure out how to assign a variable or how to do a calculation in R or how to make a function in R or how to apply a function. All this stuff is sort of, should eventually become second nature to you. But once you cross those technological barriers, then you're sort of halfway there. And then competing methodologies, and I think somebody said one time the number of methodologies to analyze single-cell RNA-seq data approaches the number of papers using single-cell RNA-seq data. So this is a very ripe area for still for people to develop software bioinformatic techniques, and that's a good thing. So there's a lot to choose from, but it's also can be daunting a little bit. And my general approach and recommendation for people who I think are in this class is to pick a methodology that works, become comfortable with it, use it, and if someone tells you, like say a peer reviewer tells you that you need to do something else, then you may have to do something else, but there's essentially no way to predict what a person is going to say in that regard. So pick one of the standard methodologies. I think what I'm going to show you here is perfectly acceptable. I've tried out a number of other methodologies, and I don't think there's a substantial difference between any of them. As long as you know what you're doing and you're using kind of staying within a certain limits, I think you're going to be safe. But again, it can be challenging. You're going to find a lot of stuff out there and ways to do things. And some of those things are going to work. Some of the things you're technically not going to be able to do with your computer for various reasons. So pick something you can do and go with it. Okay, so the first thing I want to talk about in terms of the practical application of this is, how are you going to get your data? So, you either, usually what you'll do, unless you own your own 10x machine, is you'll work with a sequencing core or a genomics core to produce these data. So they'll give you cells to the core. The core will run the assay. They'll make the libraries. They'll say it all looks good. They'll submit it for sequencing, and then maybe some conversation about exactly how much sequencing do you need, and that's a little bit of too much detail for today. But at one point, you're probably going to get FASTQ files, or you may get FASTQ files. And then you're going to be like, well, what the heck do I do with those? Because it might be like, 200 gigabytes of data or more, and you don't want to have to on your MacBook or whatever. You don't want to have to, worry about, doing all the alignment and everything else with that. So what you're going to do, and this is like definitely a strong recommendation, is to use the 10x Genomics Cloud to analyze your data. And I will show you exactly how to do that. The 10x Genomics Cloud is a free resource provided by 10x. And this is, of course, assuming that you're using 10x technology, which I think probably most people would use, and I would recommend using it. But what this is a cloud computing cluster that exists in the cloud, and you upload your sequencing data, your FASTQ files. You make select some options in terms of, what species, what reference, transcriptome reference do you want to use? What exactly was the technology that you used? Did you use, 3'? Did you use 5'? Did you make, immune profiling libraries? Did you make antibody capture libraries, et cetera, et cetera? So you fill in all those things. You upload your sequencing data. You tell it what those things are, and then you click run, and it runs. And it's going to be, orders of magnitude faster than what you could run on your home computer, or like even on my bioinformatics computer that we use. So we do not do any of the upstream processing for any of these experiments anymore. We do it all on the 10x Genomics website. Now, it has some limitations. There is as you can see here, the FASTQ files in this project have expired. So the data is only going to be stored for 90 days. And so all these data are, basically not there anymore. So we'd have to upload it again. So you can't delete the data off of your computer. You got to upload it and then use it, and then it will be deleted from the 10x Cloud after 90 days. So essentially, this is what it looks like though once you upload it. This is really basically the same thing that you would see. You upload the FASTQ files. It automatically recognizes which FASTQs belong together. It will put them all together. And you just have to tell it what type of library it was using this drop-down box. We have a question here in the chat, and definitely be asking questions. Do you need an account to use a 10x Cloud? The answer is yes. It's free though, so all this is free to use. They assume that you've spent thousands of dollars on their product. They'll give you the computer time for free. Okay. So yeah, so this is how you upload your data. And then once you've got the FASTQ files uploaded, you're going to go to, sorry, I'm gonna go back to FASTQ files again. I want to mention how to do this. You can actually, all you have to do is you can browse for files, just like, you would with any other, uploading tool. You may have issues in terms of, firewalls or something, or maybe, your browser won't handle that big a file. So what I typically will use is the command line interface, and not to be, you don't want to be intimidated by this. It's very easy to use. It gives you all these instructions down here for your operating system, Mac, Windows, Linux. It tells you how to install the program that you're going to use and what you exactly have to type into your terminal program. So this is all very easy to use. This is the major sort of time that you're going to spend doing with it is like, uploading these files, and you're going to click, then it's basically point and click for you. So The next thing, once everything is uploaded, you're going to go to analyses, and what you would do is actually there's a button. Actually, I'm sorry, you would go back here, you would select these and then click create new analysis, and then that would produce the analyses. And then you're able to go here. Once it performs the analysis, you'll click on that and you'll see this button here. It says download all output files. You click that guy and that gives you another command. Now this usually has to be done through the command line because the files are too big. But this is also very easy. All you do is you just copy these files and you type it into your terminal program, and it will just download the files for you. So that's also super easy. All right, some questions here. Even with 10x cloud to process data, you still need enough memory to store the FASTQs in the computer. Well, so memory and storage are two different things. You do need enough storage to process, to store the FASTQs and everything else. So this is not something that you want just to do on your local laptop. You're going to want to do this. And you know, we keep all of our FASTQs and all of the you know, all the output on our network drive, which has, I don't know, I think like 20 or 30 terabytes right now. So that's the recommended way to do it. You also want to have it backed up. So I would highly recommend a sort of a institutional network drive, like our OSU X drive is what I use for all of mine. And in terms of memory, the memory has to do with, you know, how the processing is operating and, you know, how the program stores data while it's processing it, and, you know, in this context. And so that has no effect because all that processing is being done on the 10x side. So do you have to upload from a local machine, or can you use FTP from a remote repository? As long as you have, I've never tried, it's a good question. I've never tried to use FTP. All I've done, you know, because usually you're going to want to have all the FASTQ files stored within your domain, okay? Because you're going to want those to be archived. So you're going to want those in some storage you have access to. I use an interactive SSH session, and I can just show you how that looks. So this is like a Linux file browser. So, you know, yours, you know, is probably going to look different from this, but I can just show you, you know, what these are. So this is my X drive here. The sequencing data happens to be in this folder, scRNA-seq vignette. Here's two samples, Chromium X, Chromium Controller. In this case, this is not always how you would necessarily have to do it. The FASTQs are in here. Okay, so here's all the FASTQs. You can see these are pretty small FASTQs. This is a sample data set provided by 10x, so these are really small. And so these are, you know, maybe only 10 gigs total for all this stuff. And then here is all the output that I've downloaded into this particular folder for that particular sample. Okay, so when you upload this, you would just go to this directory here, you know, enter your terminal or enter your shell in this directory, and then click, you know, then paste in the command that it gave you in the FASTQ uploads. You could also, I mean, this is also accessible via, and this is the same folder here, via Windows. And this is what you would select in Windows. You could drag and drop this stuff, or you could select this particular file in Windows using, you know, using this particular box here. So you can do it either way. It just depends on what sort of a machine you're using. Do you have to, that one? I've used FTP and it should work. Okay, that's good. So that's a comment here. I've used FTP and it should work. And then how to convert the cell ranger file into FASTQ file. Okay, well, that definitely can't be done because the cell ranger output is, you know, there's just no going back. Cell ranger output, I will show you now. Okay, so let me just go back since I've already shown you this this view here. This is the output that you would download from from analyses. So you would go into here, you would go to click on the first sample, you click on that thing, you would download using this code here, okay? And then you go here, and these are all those files that I just downloaded. So what is the important stuff that you want out of here? Well, the easiest approach is to look for the filtered feature barcode matrix tar file, okay? So what is that? That's an archive file, and it's got 3 files in it. It's got barcodes, features, and matrix, okay? And these are the core of what you're analyzing. And what Cell Ranger does is it takes all of the reads from your FASTQ files, it aligns them to the reference that you've selected, it identifies the cell that that read came from, it identifies the gene that that read maps to, and it identifies what's called a UMI, okay? And a UMI is like, you know, it's like a sort of like a barcode for a molecule. It's a unique molecular identifier. And this deals with PCR duplicates, more or less. And so what Cell Ranger does is it aggregates all of those UMI counts, the unique molecular identifier counts, single molecules that were captured by the bead, and it counts those up for every gene for every cell and puts that into a big matrix, okay? And your matrix is here, okay? This is the matrix, all right? And so what the heck is this? All right, a lot of numbers. And this is important for you to know, at least conceptually. The matrix that it produces is called a sparse matrix. As you probably know, 10x data doesn't capture necessarily every molecule that's being expressed by a cell. And so on average, it might capture 1,000 to 2,000 genes per cell. You know, and maybe the cell is actually expressing 5 or 6 times that number, or maybe not, but below, you know, the bottom, the lowly expressed genes are not captured as well on a single cell basis. Now, if you aggregate it, then you'll see those, but on a single cell basis, it's not the case. So this matrix ends up having a lot of holes in it, a lot of zeros, and the zeros aren't helpful. So zeros are just zeros. And in order to compress the size of this matrix down, we're only storing the non-zero values. And that's what this matrix is here. A sparse matrix is one where you're not storing the 0 or the empty values, okay? So what you need to go along with this in order to translate this into actually something useful is a list of the cells and a list of the genes, because remember, that's what Cell Ranger is doing. It's producing a list of genes produced by every cell. Okay, so we can go back here, and this is what is contained in here. It's a list of the cell barcodes. Okay, so these are all the cell barcodes that were captured. In this case, 587 cell barcodes for this sample. Okay, and this is the list of genes. This includes the ensemble gene identifier. It's a unique identifier that everyone, you know, is standard. The standard gene name, and this bit here, gene expression, tells you what type of a feature it is. So you can have multiple features for an experiment. You know, by and large, we use genes as features. We can use those interchangeably, but you may also, you know, have an antibody library that you're using, or a CRISPR guide library that you're using, and those would be different data types. And so that's a little bit more complicated, but that's what that is. Okay, so the bottom line is you can't go back, you know, from this matrix, which is a summed aggregate count number of molecules per gene per cell. You can't go back to a sequencing file. So you can't reverse engineer someone else's Cell Ranger data. Okay, hopefully that answered that. So let's go back to where we were at here. Yeah, so some other things just to point out before we move off of the 10x Cloud topic. This is a major advantage, as you're always going to have very easy options to select which Cell Ranger pipeline you want to use. You know, like let's say you analyze some data and then 6 months later you want to analyze some more data, but the version number has changed. You might want to either reanalyze the old stuff or analyze the new stuff using the older pipeline. So that's an important thing to note. I already mentioned that. Some other options that you can use is you can use the option to include reads mapping to introns, which basically is looking at pre-mRNAs, and this has been been shown to be like valid. So that's something that we're doing now, and it increases the resolution because you're going to get more usable reads that way. You can opt to make a C-loop file or to aggregate your data and then make a single C-loop file from the aggregated data. Now, this is not something that we're going to do in this class because that's an interactive program that sort of locks you in. It's not particularly reproducible. But it might be useful for people to quickly explore things interactively. You can also use the option to skip band files, which is going to decrease the size of the data that you're going to have to transfer. And I usually skip making the band files. There's for most analyses, no reason you need the band files. The other thing that this does is it generates sequencing metrics for you, and this is important to look at. It will tell you, on a nice, very concise page like this, it will tell you how many cells it's estimating that it recovered. Now, how does it define a cell? Well, what happens is, you're putting in, you're getting a lot of sequencing data from droplets that don't have any cells in them. And, this is because, the way the assay is designed, you have to have a large excess of empty droplets in order to guarantee or to be more likely that you have single cells in the droplets that do have cells. Okay, so you have to have a mathematical method for segregating the empty drops that don't have cells from the droplets that do have cells in them, and that's what's shown here. And this happens to be a very nice looking plot, which is why they're using it for their sample data. But it's essentially a rank plot of all the barcodes that were detected, and this is a log scale on the x-axis, and the number of counts that were recovered from each of those cell barcodes. And you can see it has this sort of a sharp knee here. And what the program does is it picks a spot here in this knee where at this near this inflection point to say, okay, above this, more UMI counts in this inflection point is a cell, and below it is not a cell. And then however many are above that threshold, that tells you how many cells is estimated, at least at this level, from your sample. Then, of course, it knows what reads came from those cells, and it can tell you how many reads you got and how many genes you got per cell. Now, these metrics are, really pretty extreme. The recommendation is 20,000 mean reads per cell generally, and even that I think is generous. You could probably get down to 10,000 reads per cell without having any sort of loss of resolution. But we generally calculate and try for 20,000 reads per cell. The other thing to note here is some useful information is the all this information about what version of the program. I don't know if you can see this down here, the version of Cell Ranger, the reference that you used, the chemistry that was used, these sorts of things. In here, the fraction of reads in cells, an important metric. You usually want that. They claim you want it over 70%. Usually you want it closer to 90% of reads in cells. So that's the important thing. So it's always good to scan these quality control files after you've run the sample. And I think I mentioned all that stuff. We reviewed the data. We looked at the gene barcode matrix. We looked at the gene and feature data. Okay, and here I have written metadata. We'll talk a bit more about that in a second. And now, so I want to sort of talk about the how you're going to hold this data in R. Okay, we're all familiar by now with things like a list and a data frame, okay? And a list in a data frame is what's called a, so actually I got another question here that just popped up. I'm gonna take 1 second to answer this. So how do you know if your sequencing is saturated from the plot? There is, that's a good question. So I go back here. The sequencing, if you go to analysis, sequencing saturation, that gives you like a sense for how many PCR duplicates. That's basically based on how many duplicate reads are detected in your sequencing data. And when it reaches a certain threshold, it says that it's completely saturated. I don't find that this metric particularly correlates or adds significantly more information than simply looking at the number of reads per cell. So I think, you can have, if you go below 10,000 reads per cell, you're going to start to see some differences in clusters and these sorts of things. But if you go between 10,000 and 20,000 reads per cell, you're going to have differences in saturation, but you're not going to see any differences in clustering or gene expression that I've been able to tell. So, I mean, other people might have different experiences with that, but I don't think this I don't really rely on too much. So anyway, that's a good question. Happy to have people post their experiences now or later. Okay, so let's go back here to this guy. So yeah, so we're all used to things like lists and vectors. And these are in R, and I don't want to confuse you with some terminology, but these are called S3 data structures, and they're pretty standard, pretty simple, pretty malleable, or you can sort of make them whatever you want. What we're going to talk about is an S4 data structure, okay? And the only thing you need to know about this is that they are like a list, basically, but have very specific rules attached and have very specific methods that are associated with them. Okay, so a list can be however long you want, can have whatever in it that you want, it can do whatever, you can do almost whatever you want with it. But the data structures we're going to use to hold single cell data have very strict rules about validity. Okay, and that's important because what I just showed you before was, that there's a list of genes that corresponds to the rows on that matrix of gene expression and cell barcodes. Well, if you were to delete one or more of those row names, one of those gene names from that feature list, that would cause like an indexing problem, an alignment problem. You would have like rows and data misaligned, which is like a major problem. So what this data structure, all these data structures are going to use is a mechanism to check. And so if you subset either rows, if you subset cells or you subset genes or features, it will always check to make sure that everything is in sync, okay? And so that's an important thing. Another important thing is, when you want to extract information out of this, there's specific methods to do that, and I'll show you, how to do that. Okay, and for example, here, call data of a CDS, and the CDS is, in this case, the object will return the cell metadata. So that's not a function that you can use. Call data you can't use on any list. It's specific for a particular data structure. Okay. The data structure that we're going to talk about and use for the most part in the class is what I use for my stuff in the lab, is called a cell data set. Another feature of these advanced data structures, like they're called S4 objects, is a concept called inheritance. And you don't really need to know anything more about inheritance except for the fact that the cell data set is related to another object called a single cell experiment, which itself is related to and was derived from an object called a class called a summarized experiment. Okay, and what does all this mean? All this means is Now, there are a whole family of data structures or classes that are related and have shared and inherited rules and methods. Okay, so what this means for you is that there's a whole family of packages that are based on these methods that you have available to you to use. And these are specifically bioconductor. Okay, now you should be at this point familiar with bioconductor. If not, bioconductor is the largest open source reference for packages for codes and data that you're going to use for genomic analysis, for transcriptomic analysis, for sort of anything biology related. It's going to be in bioconductor. And so having what you're using, the cell data set, be based on a bioconductor data structure is a good thing. Okay, it allows you to use many more functions than if you were to create a new cell data set or a new object, a new class of object on your own. Okay, so having that related is a good thing. Now, this is different from Seurat. So sorry, they're like drilling here in my office. So Seurat is another package you're probably familiar with. And in fact, it's a very popular package for analyzing single cell data. The Seurat data object is different from the cell data set. Okay, it doesn't inherit anything from the cell data set. It has similar features, but it's not the same. So if you're going to want to use Seurat to analyze your data, you're not going to be able to use directly any of the bioconductor functions, and vice versa. So if you're going to use a cell data set, you can't directly use the Seurat functions. And so you have to have some code or some ability to translate between the two. And this ability exists. I've built a number of functions that will convert from cell data set to Seurat. And Seurat has a number of functions that go the opposite direction. And so what you're going to want to do is sort of, and I'm not here to say like one is necessarily better than the other, but you should know the limitations of each. There are also many packages out there that are built directly off the Seurat object format. So my recommendation is to basically pick one and then become familiar with the ways to convert between the two when you feel like you need to convert between the two. I prefer to use the cell data set objects, but that's really an opinion rather than a strong statement of truth. Yeah, so that's my comment on methodology and data structures. Okay, so we are going to now go through a few more things, and I want to bring up my R Studio now, and I want to mention a few things about analyzing the data. Okay, so first thing to mention is that we have a new option for installing packages that we use, and I don't want to take too much time to do this, but I just want to mention that we have a new repository which will help you access these packages more quickly and more concisely. And if you are interested in using this, what you should do is you should run this command here, use this edit our profile. That will bring your our profile document up so that you can add these lines to it at the bottom. And that will activate the custom features and custom functions that we're using here. So you put that in there, and then once you restart your R, you'll be able to access a couple of things, namely here it is here, there's my face, lovely, namely Blazor templates, which you should be all familiar with, but more importantly for today, Blazor tools. Okay, and Blazor tools has a lot of complicated functions for analyzing single cell data, and it's a lot faster to download it and install it from this particular mechanism, which is why I'm mentioning it. So that's the recommended way to go for that. You're going to want to install Blazor tools in order to do what we're going to do today. Okay. Yeah, so let's look at dependencies. All right, so you're going to want to edit your dependencies, and this is in the workshop project, and these all will be pushed up to GitHub for you to take a look at. But there's some changes I want to mention. We've installed Blazor tools, and so you're going to want to do that. You have to attach it using this command here, library, Blazor tools. And you're also going to want to put these in your dependencies file, the Monaco 3 and Seurat. We need to have those attached for certain functions that we use. Okay, so those are some changes there. The other thing to point out, and it will tell you to do this if you don't do it, but there's a conflict in some of these in one function that you're going to have to attach called row ranges, and you have to add this line here. So if you forget to do that, it will just tell you what to do, and you have to put this with your other conflict statements here. Okay, so I've already sourced all this stuff and run it, so it's all good to go. But you're going to want to remember to source dependencies and configs, and then you're good to go. Okay. So I mentioned that. Okay, so now let's sort of walk through the analysis. Okay, and I've put here actually another thing. So this is going to load the template for you. All right, so load process to R. All right. So this is basically a script that you can more or less configure and then run it. And I would recommend when you're starting to run it interactively. And the reason is that you're going to want to maybe change things. Some things might be not configured correctly. It's a pretty complex taken as a whole. And so you might want to look at the data objects as you go through. So my recommendation is that you run this interactively. Eventually, once you've run through it once, it should be set up to the point where you can source it and run it all again, so that it's all clean, as I've recommended previously. Yeah, so in any event, I can tell you that I just ran this a few minutes ago before we started, and this thing will run on a desktop computer in about 10-15 minutes. So some of the steps do take a little bit longer, and if you have a lot of cells that you're looking at, it's going to take quite a bit longer. Okay, so let's sort of walk through this. The first thing that you're going to want to do is to load a configuration file. And what this is going to do is going to carry all the information that you know pre-existing about your samples. Okay, and I'll just run that, and that didn't work. On that, okay, and so that's the analysis configs. And if I call up analysis configs, I'll show you exactly what's on there. It lists the sample, and this is the same, basically the sample name that we got, that we used when we generated the 10x data. Has the date the sample was collected, and has the equipment, just another name for the machine that the sample was run on, and has a path, a file path to the like the enclosing folder. Okay, that's the folder I showed you here. Okay, the file path to this folder. Now you might notice, I can just show you in this form here. This is the same exact file that was read in. You notice it's got this sort of weird, // file path. You're going to have to configure the file path depending upon your system. So if you're running on Linux, it's going to look different from Windows, and it's probably going to look different from Mac, or it might look different from Mac. And it all depends on where you're storing your data, what computer you're running it on. So this is this is configured for Windows this way. So that's going to be a very frustrating situation for you, but you just have to figure it out. Okay, now of course you would put anything else in here that you wanted. In this case, the data set is just comparing two methods of capturing the data, either the Chromium standard machine or the Chromium X machine. Okay, and yeah, so but you could put anything else you want in here, like date, or if you're doing patient samples, the patient sample, patient ID, patient features, clinical data, mouse experiments, you put mouse treatments, time points, anything else you want to put in there about a sample, a biological sample would go in this as a separate column in this file. Okay. And what's going to happen is the program that we're going to run is going to add all of those into our data structure as additional cell metadata. Okay. So you'll see how that goes. The next thing that I do is I sort of grab all of the sequencing quality control data that matters, okay? And I put that into a file. In this case, we're calling it seqc. And this is a, this just takes those, these are the same numbers that I showed you on the website from before, estimate, but it's only pulling the ones that I think are important. The estimated number of cells, reads per cell, genes per cell, and fraction of reads per cell. Okay, so this is a table that you can plot, or you can just hang on to it or print it out, but that's the quality control metrics that I think are most important. All right, so that's seqc. That should all run pretty automatically if you've got the files identified correctly. Okay, so the first thing that you're going to do is you're going to run a program, you're going to run a function, more or less, to grab those barcode matrices and put them into the data structure that we talked about. All right, and that's what all this code does. And you can see here the first thing that it says is map. And you all remember what map is. Map takes a list of objects and then runs a function using those list of objects as input, and then returns a list of objects. Okay, so what we're going to do is we're going to take all the samples, and then we're going to grab the matrices and put them into a CDS or cell data set object, and that's what this function does here. You can ignore this for now, and then it's going to just return those. And what it's going to do, it's going to return to the object CDS list. Okay, and so what is this exactly? So let's look here, okay, at week 4, and I'll put this up here so you can explore the things on your own when you if you want to look at this. So CDS list, you can just that, and you can see it's a list of 2 objects. The first one is Chromium controller. The second one is Chromium X. So you have one item for each sample. So that's good. It's a cell data set class. dim tells you how many genes are in each cell data set. In this case, 601, and how many cells. The second number is always cell. So first number is genes, second number is cells. Has some other information in there. It has just a sort of synopsis, or the sort of the beginning of the what's called the row data or the call data. Okay, we'll explore those in a minute. And it has some other what are called slots for holding additional information about your samples. And we'll get to those later. Okay, so now you have 2 separate CDS objects, and they've been returned as a list. Okay, good. So, and you can look at each one individually. That will give you the first one. Yeah, so the other thing to look at is the call data. All right, so what is call data? Call data is cell metadata. It's everything that we know or that we calculate about a cell, a feature of about the cell. So if I run this, it's going to give me a table, a data frame. Okay. In this data frame, there's 705 rows. That's 1 row for every cell. Here's the cell barcode. Here's another, and that's the row name. It's got that as a separate column there. It has a size factor. Size factor is the sort of a normalization value that's used to normalize gene expression values. Okay, it's got the other information that we put in our configuration file. The date, as you remember, was in there, and the equipment was in there. Okay, so it's the function has added all that into the object, and you'll remember that size factor wasn't there, date wasn't there, equipment wasn't there when we looked at the raw data that was output from the 10x. All right, so this is calculated on a per cell basis, and this is added. Okay. All right, so the next thing we can show you is row data. All right, so you'll see that this actually was stuff that was already generated when we looked at the 10x output. The gene short name that was already there, the data type was already there, and we already talked about gene expression. Okay, so these are all of the features, all the things that we know about each of the genes or each of the features. Okay, and then the third thing to point out is really the sort of the crux of the matter is the expression matrix, and this is held within the object as well. So we can access that using this command here, exprs, and that gives you, as I discussed, a sparse matrix, okay? Here, where each column is a cell and each row is a feature, okay? And you would have a row for each gene. So there's 36,601 rows here. Okay, so that is basically what's within this object. Now Seurat has basically a similar idea. It's just organized differently. All right. The next thing that we do typically is we've already done sequencing quality control, but we want to identify poor quality cells. So if a cell is damaged or if a cell is just, whatever reason is dying, that cell is the RNA is going to be degraded from that cell. The mRNA is going to be degraded, and the mitochondria become leaky, and all the RNA that's inside the mitochondria is going to leak out, and so you're going to end up having a higher percentage of mitochondrial RNA in each cell and a lower percentage of detected genes, or a lower number of detected genes. All right, and so we use both of those features to identify poor quality cells, and then we do sort of an adaptive approach to exclude the outliers. Okay, so we don't set a hard cutoff at any certain number, but we exclude sort of the outliers, more or less. And so we can take a look at that. And what this function does is it goes through each of the CDS objects in CDS list. It runs this function here, QC, BBQC, and then returns a list of objects. And what are those? Okay, the first and probably the most important, the first and probably the most important is this, which is a small table of each barcode and whether or not it's been flagged by the QC algorithm. Okay, and if false here is a good thing, that false means that it hasn't been flagged, which means it's a good cell. Okay, so that's the first thing that comes back. And then it's got some plots, and you might want to, for example, inspect these plots and see, all right, what is the quality of my sample overall? And maybe like if you're running 6 samples or 8 samples, and one of them is like totally different from the others, even if you think you can like filter out some cells, you might just want to throw out that whole sample. Okay, and so I'll actually start here by showing you this plot here. It's a violin plot showing the log 10 of number of genes detected, and you can see below this line those cells have been flagged as being poor quality. Okay. The other thing we can do is we can look at the mitochondrial percentage, and here it's 13%. That's kind of on the high side. Usually like to see less than 10%, but it's not the end of the world. So anything over 13% has been cut off from the sample. And you can see here before thresholding and after thresholding the cells that you have. Okay. This is the sort of the summary. This one is the summary, what it looks like for all this, after both factors have been taken into account, the mitochondrial and the reads detected. So that's the cells that you're using that have passed both filters. The last 2 objects you can see here are the actual cutoffs that we've used for these features. Okay, and then so this is something that I would usually save, because this might be something that, for example, a reviewer might want to know, like what were the cutoffs that were used, or why is this sample so different? Is it, was it a poor quality sample? So this is something that's usually good to save. All right. The next few things, these are, all sort of come together here for this particular function called doublet finder. All right. And so this will be the last thing that we're going to talk about today, and then we'll pick up here next week. What doublet finder is a program that basically generates pseudo doublets by taking random cells, combining them at various thresholds or various levels in various ways, and then using that as a reference and then mapping the actual data onto these sort of synthetic doublets, okay? And if a cell maps onto a synthetic doublet with a degree of certainty above that degree of, above a threshold of certainty, that will get flagged as a potential doublet, okay? And it operates, a couple things to know about this, it operates on two different thresholds. You can have like a low confidence and high confidence doublets. So it'll give you two different types of doublets. And so this is something that you can choose. You can choose not to do it. You can choose to keep or to throw out the low confidence doublets or throw out the high confidence doublets, okay? So this function takes a while to run. It's one of the longer functions in this whole script. On this very small data set, it took about 5 minutes, I'd say. So, if you're doing 100,000 cells, it might take 8 hours to run. So this is a long running function, and you might not be able to run it on your MacBook. So just so you're aware, but you might be able to run it. Just run it overnight or over the weekend. Okay, so what does this return? So if we run this, we can explore the doublet finder list. Okay, and it's very simple. It's a list of 2 tables with cell barcodes and whether or not they're a singlet or in this case a doublet. Okay, and you can see you have columns for low confidence and high confidence. Okay. All right, so actually I'll just talk about these last 2 items here. So both these functions are pretty quick actually. So you've now got a list of the original data objects. You've got a list of the QC data, and you've got a list of the doublet finder data. What you want to do now is you want to rejoin all these back together so that we can eliminate the poor quality cells and eliminate the doublets if we want to do that, okay? And so you have to put all that information together in a list, and then you have to join all those objects as a list. You have to join those into a single CDS, and that's what these functions do here, okay? So by the time we get to what's written here, CDS main 1, we've gotten all of the data together, and I will just show you that. We got all the data together with all this useful information. We'll just run this again. We've got the cell ID, so the cell barcode, which sample it came from, that's the unique ID for the cell. You got the individual barcode without that other information about the sample in there. Size, factor, date, and equipment. You remember we started with that information. You've got the QC calls, you've got the doublet finder low and high confidence calls, and you've got the sample identifier here. Okay, so you've got these are all the cell metadata information that you're going to need to get started, and it's all in a single object with 2 samples in this case. Okay, and so where we'll go from here is doing all the rest of the stuff for single cell. We'll do it next week and then maybe into the final week of the course. Okay, I'm just going to answer any additional questions that have come up. Okay, someone's asking what's conflict again. Not sure exactly what context you're asking about conflict. You can have conflicts in terms of like cell barcode conflicts. So if you have 2 samples and it's possible that, you get the same barcode from each sample in your data object, which is why when you're putting these things together, they need to have this unique sample identifier on there. So I'm not sure if that answers your question. If not, you can email me. And then is the config file that output you get from 10, okay, is the config file that output file you got from 10x Cell Ranger with the UMI and barcode in it? So I'm gonna put this up here so people can read it. No, you have to make the config file, all right? So that's an important point. You can, I'll include an example, the example that we're using today. I'll link it in the course materials. But you have to make that. There's only 2 things that are important on there. Actually, you need to have 3 things in there. You need to have the sample identifier. You need to have the path to the 10x output, and you need to have one other metadata feature in it. And that's sort of like a bug in the program that I've written, that you have to have an additional column in there, but usually you're going to have a lot of different columns in there about your samples. So, but you have to make that. It needs to be a CSV file, and the hardest part is correctly formatting the file path to the actual data. Okay. All right. So I think that's okay. Here's another last question. How could we obtain example FASTQ files to run the analysis? So if you want to do that, you can get them from, I'll put a link to it. You can get them from 10x. You just have to download them. All right. All right. All right. I'll try and answer all the rest of these later. It's 11 o'clock. Thanks for joining, everyone, and we'll see you next week.